All right, well, while we wait for a few more people to show up, if I could, a quick survey of the types of people that we have in the audience, try and make it a little more relevant as we go. Quick show of hands. Yes, maybe who would consider themselves maybe kind of software developer as your role? Okay, couple. What about kind of bioinformatician or kind of more statistical analysis? Maybe project management or kind of research coordinator? Okay. Anyone? I think that's pretty much all hands. Anyone who didn't raise their hand? Kind of shout out kind of their roles. Okay. Okay. Great.
All right, so when we get started, so so thank you everybody for coming up to this event. I work for the Wisconsin National Primate Research Center, that is down the street. My name is Daniel Nicolalde. We maintain an instance of LabKey server to manage all the electronic health record data from our monkeys. And we built this system on top of LabKey. They were coming in town to start another project that we're interested in developing to manage the finance of our center. So since they were already coming into town, I worked with Isabel Gerard to set up a presentation and also an informal Q&A session that will be in the later half of this 2 hours. So I'm hoping that you guys brought some questions, some concerns, interest on using LabKey. So without further ado, I would like to introduce Josh Eckels. I've been working with him for better 5-6 years now.
Something like that, yeah.
Yeah, so I started with Adobe PRC right when we set up the EHR system to manage all our monkey data. And so since we maintain a support contract with LabKey, and it's been great to work with them, add new features to the system, incorporate some of the user feedback, things that they want to see in LabKey, and bring it to Josh, and he has been able to guide us through how we can expand the system. So he's the Vice President of Engineering over at LabKey, and he's based in San Diego, and he's natively from Wisconsin. So
that's right. Grew up close to Waukesha, so always nice to be back. All right, thanks, Daniel. So even though we've kind of officially partitioned today into kind of the more presentation section first half, and kind of more ad hoc Q&A after, please feel free to jump in as I go if you have questions. There might be some stuff that makes sense to table till a little bit later. Kind of be a little more hands-on specific features or things like that. But maybe Ben, you can make a note and as we go, things that we want to loop back on. But want to make this as interactive as possible. Don't just want to be up here talking at you guys. So let me know what you're most interested in. And Daniel did pass along some of the questions or areas that people had indicated they were interested in. So I'll try and hit those as we go, but please drill in anything you want more detail on. And then also like to introduce Binal Patel, who's here in the front. She's also at LabKey. She's a developer working on primarily the electronic health record systems for both the Wisconsin Center and the center at UC Davis in California. So kind of overall, structure-wise, a little background on kind of LabKey, the software, kind of LabKey, the company, and how we operate. Talk about kind of some of the high-level things that different groups are doing with LabKey Server, and then for maybe the second half of the slide part, dive in a little bit more into kind of specific features or use cases to get a little more specific. So this is kind of LabKey, the company. So the initial software started out as a proteomics data management system. at Fred Hutch Cancer Research Center in Seattle. So the project really started in one of the labs there, and then as other groups got interested in using the software, LabKey spun off as a separate but still affiliated company because that single lab wasn't really interested in building out flow cytometry features or adding a lot of documentation or support. So it's been about 15 years or so of kind of ongoing development. As Daniel mentioned, Binnell and I are both in our San Diego office. Main office is in Seattle, actually kind of right across the street from the Hutch for those who have been there. And we're kind of 45, 50 people at this point. And we've got a lot of different kind of deployments of LabKey server, kind of spread out geographically, mostly in the US, but sprinkled throughout Europe and elsewhere as well. In terms of the kind of major themes of how we approach these problems. So LabKey server is open source. We have our community edition, which is free to download and use, and you never have to talk to us about it. We hope you do, but there's no obligation to engage with us. We use the Apache 2.0 license, so pretty liberal. So kind of take it, do whatever you want, resell it to someone else if you feel like it, and then we're on hand to provide services, support, etc. If groups want it. We're trying to be pretty modular, be able to develop components for individual groups to address their specific needs, but also have some core features that hopefully can be leveraged in a variety of different scenarios and use cases. Yeah, the rest is kind of general software overview stuff. So the kind of quick kind of system diagram. So we've got a whole bunch of different data sources that you might be collecting in whatever it is your research has. Those might be relatively unstructured things. You've got a PowerPoint presentation from a lab meeting. You've got a PDF with your SOP for some particular wet lab process you're doing. You may have more structured data that's either in database form or just a bunch of Excel files. As I've seen over and over, Excel is by far the #1 bioinformatics tool in the world, which is kind of painful and scary in some cases. And you may be integrating with other existing systems, so maybe a full-blown lens, maybe particular analysis software from an instrument vendor or open source package. And you may be getting specimen data, assay data, clinical demographic data. Sometimes really you just want to look at one of those pieces kind of in isolation, and maybe you're doing really high throughput analysis, running thousands of samples over a year. Maybe you're trying to integrate across different assay types or so forth. And of course, since it's research, the types of data are always changing. It's not that there's one fixed set of proteomics data or any of these different things. It's kind of a moving target. Everyone's doing their own kind of unique thing on that. And then you want to have kind of these collaborative workflows that you can build on top of that. So maybe you start just with your lab, maybe you want to share data with a collaborator, give them access to some slice of it. Maybe they can upload data, maybe they can only view it. Maybe some of it you're supporting a publication and you want to share that publicly to just kind of anonymous users on the internet. And a lot of times, of course, that's now built into the grant requirements that there be some sort of data sharing provision. Usually it tends to be pretty fuzzy. Some specific fields are a lot more focused on you have to deposit into this repository or things like that. And then we're kind of in the middle of all of this. So adding security, adding auditing, other pieces on top of kind of that raw data, and then doing some analysis in LabKey server itself, reporting, visualization, etc. And a lot of what we're doing is kind of trying to keep that data organized so that you can do your own analysis on top of it. Our background is really more on the computer science, computer engineering side, less on the biology, bioinformatics side. And so a lot of what we're doing is collaborating with groups to organize their data, let them run their own tools on it, for example, as opposed to us being the group that produces that end result ourselves. So, kind of high level, marketing type points before we kind of pivot and talk a little bit more about specific features a little bit later on. So, that data integration aspect. So you've got disparate data sources. Maybe they're all coming from the same group generating it. Maybe it's from a single lab, but you want to be able to link those together. And maybe you're collecting data on a set of subjects over multiple time points. And whether those subjects are humans in clinical or observational trial, whether they're yeast cell lines or primates at the primate center. A lot of the types of integration that you want to do are pretty similar. The centralized data storage, so we've seen a lot of times when folks are starting with, say, Excel or similar, you've got a bunch of files emailed around, maybe you've got a file share, maybe you've got a postdoc who has a lot of stuff on their laptop and they complete their 2-year stint, and who knows where that data went or how to make sense of it. Kind of what's the context of that? What's the metadata connected with those results? So those are some of the big kind of data management challenges we're helping groups do. Trying to have repeatable workflows on top of this. So kind of bridging that gap between, okay, we're still trying to figure out what our analysis looks like, and we're doing it very ad hoc, get that kind of worked out, and then now we're ready to scale up and run hundreds of samples. So getting that workflow where you can repeat that, you kind of turn the crank and have some confidence that the workflow you did on sample 200 is the same as sample 1. Being able to have a spot to run QC on the data. So this is, I'd say, kind of a growing area for us. It's been a major focus for some of the groups that we've worked with. It's one thing to collect the data, but it's another to have confidence and then kind of a workflow on top of that. Okay, I've got my private area where I import that data, do some QC on it. Okay, now that's ready to share with the wider group. So trying to both have the tools in place to do that validation itself, and then kind of a mechanism to integrate with that with a larger whole, but not have it be kind of a constant live stream that may or may not be dependable quite yet. Being able to drill down kind of data provenance, you've got a summary view, and maybe, or you want to go back to the original, analysis results back to the raw data. Maybe you've got sample derivation protocols where you're tracking, okay, we've got a blood draw from one of the animals, and then you're doing enrichment on that, and then you're doing PCR, and then you're doing other preparation. Being able to trace that lineage and figure out, okay, where did this come from, who did it, when, and so forth. Visualization analysis, I already touched on a little bit, and the secure collaboration. So this is really a key area for us, is making sure that people have ways to get kind of the right granularity of control over their data, that you can have these different slices that different groups can see or be able to modify or upload, and having some of the auditing controls on top of that, so that once it's been imported, it hasn't been modified, or if it has been modified, what was that modification? Because of course it's rare to have everything right exactly the first time on import. So kind of switch a little bit and talk about some of the example installations, and some at the center here, or at the university here, and then some at other groups. Try and get a little bit of a sense of kind of the breadth from kind of individual labs, kind of working with it, maybe a PI and 10 folks in the lab, to data coordinating centers, to full-on kind of multi-organization, multi-continent consortia, trying to organize and share their data. I'd say most of the users of LabKey server are somewhere in the nonprofit realm. A lot are based in universities or similar, but we've got, I'd say, a growing base in biotech and pharma, especially in some of the new areas that we're pushing into. Yes, let me pause there. Any questions? All right, keep going. So I think I've got maybe 4 or 5, kind of different groups highlighted, kind of give you a sense of the high-level things they're trying to accomplish, and then, like I said, loop back with a little lower-level feature discussion. So immune tolerance network. So this is one of those kind of larger research consortia. Really, their focus, as you can probably guess from their name, is kind of an immune reaction to various things. So transplant and kind of immune suppression drugs. One of their major studies was, okay, how can we taper transplant recipients off at least to lower levels of those doses to reduce the adverse side effects without increasing the risk of rejection. So they've been working very hard at not just their internal analysis, kind of using LabKey to manage that data and come to their publications and findings, but also to expose the full backing data for all of the papers and all the clinical trials. And there's a publication of about this a while ago. I think they were basically one of the first groups to kind of do that full set of raw data sharing. Of course, that seems to be kind of a trend with kind of recent concerns on some of the other trials. But they actually went kind of a step further, and not only did they say, okay, here's the raw files, here's our analysis results, and here's the paper with the final figures. They took the extra step of saying, okay, here's the R script, for example, that we used to come up with this figure, this result. And here's a user interface where you can actually tweak some of the parameters to that. So you can kind of do your own interactive analysis on top of that. And okay, if we do vary or kind of slice the cohorts differently, how does that affect the output? Obviously that itself is a major effort, kind of trying to make that not only something they can use internally, but something that someone else external has some chance of coming up to and exploring in a meaningful way. But I think has been a very cool project. So they've got a whole bunch of investigators across these different studies, a lot of concurrent clinical trials in process. Second example, so one very near and dear to Daniel. So this is the electronic health record system for national primate research centers. So basically, we started this project with the Wisconsin Center here. That was something like 8 years ago, a little before you had started. And what we did is we took what we call our study module, which collects data for different subjects across time points and helps integrate that so that you can say, okay, where is this animal housed right now? What is its most recent weight, its sex, species, and when did it have its last TB check? And so being able to kind of integrate these into single views, capture that data. So this is their primary clinical record system. So absolutely kind of mission critical for them. And then our hope from the beginning was to be able to roll that out to other groups, and that's kind of a common theme here. As we add new capabilities to LabKey server, we want to make these features available to the community, hopefully leverageable, but at the same time, kind of focus on what do we need to get this thing working first, and then kind of expand later. And so I'd say maybe 2-3 years after starting development, so maybe a year or so after rolling it out, replacing the existing center, the existing system at the center, the Oregon National Primate Research Center said, we're interested in replacing our system as well. And we kind of took that core EHR that Wisconsin had developed and made it possible to customize it for each center, because and this is very common, I think, as other groups adapt other areas of LabKey. There's kind of maybe 80% overlap between these different groups and the terms they use for a particular thing and the workflow they have and the data they're capturing, but it is certainly not 100% aligned. And so making it so that there's kind of that core common base and hooks to customize for the different groups. And so since then, we've been working with 3 of the other centers to kind of adapt and roll out for them as well. So that's been great. And given kind of the open source approach, everything that either we develop with one of those centers or their own developers like Daniel and his team develop are then available for the larger community, but it's kind of an opt-in approach. So, we can integrate that new piece, but we don't necessarily have to if it doesn't fit with our needs or workflows. So kind of on the larger scale genetic side, there's a group called Genomics England, as you can probably guess, based over in the UK. And they have this pretty ambitious goal of doing full sequencing, 100,000 human genomes. They're very integrated with the NHS, National Health System, over there. They have a bunch of, I think there might actually be more than 12 at this point, but a bunch of kind of key hospitals, they call them genomic centers, in the NHS over there. And so really, their goal is to be able to focus on these kind of rare diseases and build these kind of family pedigree, look for sequence homology across different individuals there. But our role in that is more to help them organize kind of the metadata. So we're not directly involved in doing the sequence analysis. They already had some existing systems, in-house expertise they wanted to continue to leverage there. So basically, LabKey is receiving data from all these different centers about kind of demographics or clinical history for the individuals, and then kind of organizing that, giving them a portal to say, okay, how many patients do we have in the system with these various characteristics, and then be able to link that back to their sequencing data that's maintained outside of LabKey. Immune space and other kind of general research consortia, they have a variety of different studies that they're using LabKey to kind of back access to that. So it's both available publicly and to individuals kind of within the overall consortium. Get back to the raw data. So again, kind of it's similar to that first case I highlighted, and a trial share, being able to kind of link the raw data through the analysis and kind of drive towards more reproducible research results. And they make heavy use of R as well. Knitter is kind of a pretty popular R package for doing some of these visualizations and combining kind of the text description of what you're doing and kind of the code that's generating the results. Another example here at the university is Dave O'Connor's lab, so AVRL, the AIDS Vaccine Research Lab, kind of over in the research park. So we've been collaborating with Dave for actually even longer than the Primate Center. He kind of connected us with them. So he has a system in his LabKey installation where they manage data for, right now they're up to, I think, 19,100 and some experiments. Those are wildly varying granularity. Some of them are, okay, I kind of did this one plate-based assay on this one sample. Some of them are much bigger. It does, so he's very involved in the Primate Center, affiliated with them, and so a lot of the data he's generating is through research on animals in the colony, and so there's integrations from the system that Daniel maintains to pull information over to the lab installation. They're doing quite a bit of genotyping and sequencing, real focus on MHC typing and the allele identification there. Across a variety of different sequencing platforms that kind of kept moving across as the technology advances. And then one of the most exciting things that we've been doing with them over the past year or so is doing real-time data sharing on some of their Zika research. So Dave was pretty early on. He's got some collaborations with folks in Brazil, and so had connections kind of on the ground early on. And what they're doing is publishing data ahead or sharing data ahead of publication. So almost kind of day of, as they get new viral loads or flow cytometry results, they are uploading that, and you can kind of chart these the course of these experiments, kind of on a day-by-day basis. They've got, I think maybe up to 20 kind of different studies as they've organized them in varying stages of completion. And so that's publicly available. There's kind of a private area for the lab where they're uploading, kind of doing their own QC, and then kind of exposing that to the general community. So that's at zika.labkey.com if anyone wants to take a look. That it's an open portal, so there's some other groups sharing results as well. I think O'Connor Lab is by far the kind of the most established with that, but it's kind of a resource we've tried to make available just to the research community overall, specifically around kind of this more real-time release of data. That's kind of the end of that section on installations. Again, pause for any questions, comments on those? All right. So a little bit of a pivot now to kind of a more kind of technical or lower level details. So talking about the kind of basic deployment architecture of LabKey server. So the core server itself is written in Java. It's a web application, so pretty much all access to it is over HTTP, whether that's user interacting through a browser or API calls from a variety of different client languages. We use Tomcat as our web server. So again, trying to make sure we have a full open source stack that we can deploy on. We use either Postgres or SQL Server as the primary data store. That's the one that LabKey manages and is importing data into. We can connect to other data sources as well, Oracle or MySQL, to expose data or to transfer data in or out. But as far as kind of the core backend, Postgres and SQL Server are our supported platforms. And then we have what we call modules, pretty similar to a lot of other software packages that have some set of functionality that can be database schemas and user interface and analysis scripts, kind of packaged together around some kind of discrete useful unit. And so generally they can kind of contain a mix of things, depending on what you're trying to accomplish. You can have Java code, but you don't need to. SQL queries, a lot of what the modules are doing is kind of managing data that ends up in that relational database, and also a real focus on user interface. So built-in LabKey views and very specific customizations on top of that for particular workflows. At a very high level, I kind of broke down LabKey server features into 6 or 7 kind of main themes. So I'm trying to touch on all these. I know that some of these align with things people express specific interest with in the questionnaire. So assays, kind of either raw or analyzed results from them, specimens and samples. I always kind of say, goo goes in, numbers come out. Kind of all the goo and where that came from. Data integration, so connecting these kind of disparate data types in some sort of meaningful way. Collaboration tools, so a lot of what we've seen groups need is help not only managing the data, but some of the communication around it, some of the context, or just a place to have a wiki or a discussion forum. Security and permission, so making sure the right people have access to that data, but not more than they should. Kind of reporting, charting, ways to get data out in different formats. Excuse me, and then connectivity with external systems. So I know someone mentioned RedCap in their comment. So connecting to other packages, other software, because we're very much in how can we mix and match with other tools that are already working. As much as a software developer, I'd love to be able to build whatever. You can't always build a better whatever than someone else is already using. So how can we connect to it and complement it? Assays. So like I said, we kind of got our start early on in proteomics. So for those familiar with kind of the mass spec world, protein database search using tools like Xtandem or Mascot or Sequest. More recently, we've been doing a lot of work in targeted mass spec. So instead of kind of biomarker discovery, more kind of quantitative results. So, okay, I've identified biomarkers of interest, and I want to really be quantitative about those. And I've narrowed my list. So instead of doing that kind of wide net, okay, here's my 100 or 1,000 proteins that I know are of specific interest. Flow cytometry was a kind of early addition on the assay side. So FlowJo is really a tool that we integrate a lot with. Kind of desktop application, people set up their analysis and import those results in a LabKey to share, combine, and integrate with other data. A lot of simple plate-based assays. They tend to be lower volume in terms of the output, but a lot of times far from simple in terms of what someone's actually trying to do with them. So a lot of the kind of standard ones that we've worked with, ELISA, Luminex, et cetera, and then some really kind of custom ones that we've created for different groups. There's a neutralizing antibody assay that kind of started out a lab doing basically a bunch of Excel macros. And so they'd have, I forget how many they were up to before they started importing into the LabKey server, but somewhere in the 1,000 to 2,000 range, I'd say. And they've gone through a number of pains where they need to update that macro because, okay, now we want to do a 5-parameter fit instead of a 4-parameter fit. And that's not very convenient to modify that many Excel files and have confidence you're doing the right thing. So at this point, they're up to, I think, 60 or 70,000 kind of discrete runs. So this is HIV and kind of vaccine work that was driving that, but then basically a number of their collaborators started wanting to run that same assay, and they were able to use that same kind of analysis result, which is great. And then this is one of the main areas that we're adding new capabilities for different groups because either they're working with a new file format that we haven't directly supported already, or they want to connect to some other existing analysis software and be able to run that and import those results. And I should say there's kind of a lot of variation in terms of how much data we import for any of these different things. Sometimes it's really more just strict metadata management. So for some of the sequencing work that we've been doing with the O'Connor lab, really all we're doing is kind of tracking the samples, tracking some of the kind of sample prep, and then pointing to, say, the FASTQ output files, but not actually importing any of those reads into the database because that doesn't provide a lot of value. Whereas with some of these plate-based assays, where essentially we have 96 rows in a spreadsheet, more or less, one for each well, okay, we'll import those full results because there's more interesting things you can do with that, and the data volume is just a lot more manageable. We have also what we call our general purpose assay. And so this is a way to be able to import from an assay, kind of keep track of that data, but not necessarily need to have direct support for that assay in LabKey. So it's more kind of a generic import. If you have, say, a TSV or an Excel file with kind of predictable header columns, we can go ahead and configure what we call an assay design. So an administrator sets up, says, okay, I'm expecting time, subject ID, temperature, and whatever the value from the instrument itself. Maybe it's the fluorescence, and be able to slurp that up. We can also do some scripts in front of that, so that if you have kind of a non-standard format, we can kind of transform it to kind of a rectangular grid and import that. Or if you want to do some analysis before getting it in, so it's not just the raw data. And this is great because it's very flexible, right? You can just kind of configure, kind of get up and running quickly. And a lot of times that's where groups will start. And they say, okay, great, now I want to add this more complex analysis or this kind of meta-analysis. So across all these 100 or 1,000 samples imported as discrete runs, let's do a normalization pass on those. So kind of that downstream workflow. And so we can kind of build on top of that basic spot. But this is a great way to just kind of, okay, I've got this assay data. How do I get it in without having to do kind of a ton of customization up front? Specimens and samples. So there's kind of two main modes that the people are using us for in this context. So first, I would not classify LabKey as a full-blown limb system, whatever that means. Generally, we're not doing kind of freezer management or kind of very detailed sample tracking like that. So if you're using us as your primary database for samples, generally it tends to be kind of lower volume. That you don't have 100,000 or 1 million samples. That you're more in the kind of 1,000, 10,000 sample range. And so we've got capabilities to enter that directly into LabKey, or if you are running kind of one of these much bigger projects and you want to know exactly what rack and cane that sample is in the freezer and see a visualization of that, odds are you probably already have some other limb system, and so a lot of times we've set up connections to export from it and import into LabKey on a nightly basis or weekly or whatever frequency makes sense. We can track that sample lineage, so kind of that derivation process. What is the provenance of a given sample? And then we also have some tools to do what we call specimen request workflow. So for these bigger consortia, they've got 100,000 samples, maybe spread across multiple sites. We're getting kind of a feed of those coming in to LabKey nightly, weekly, whatever. And then they have investigators who want to use those samples, do some sort of assay on them. And typically, there are, of those 100,000 samples, some much more interesting samples from select group of participants. Maybe for HIV study, it's samples right around the time point where someone got seroconverted, and so that's a really interesting time to see before and after. But those are very valuable samples then, so you don't just want to say, sure, everyone grab them in the first 2 months of our study, and now they're all gone. So having a specimen request coordinator who kind of approves those, curates them, says, okay, yep, please send those over there. We get the new limbs feed. Okay, now we know those have been received. Okay, now we get assay results and kind of build that full workflow on top of that. Data integration. So this is something that we manage in what we call our study module. So this is kind of the basis for the EHR system at the Primate Center. This is really organized around the idea that you've got a set of subjects and you're collecting a variety of different things across different time points. Maybe you do multiple things at a given time point. Maybe that's a human participant in a study coming into the clinic, filling out a questionnaire. How are you feeling? Okay, let's get some basic vitals on them, and let's collect a sample and then assay that later. And so kind of being able to integrate and pull together in some reasonably automated way. For this time point, what were the various values at these other data types? And it also has some capabilities to collect data from an assay. So, okay, I've run my Luminex assay, for example. I've got my plate results. I load that to my private kind of lab area. I do some QC on that. That looks good. Now I can selectively copy, say, the 20 samples that belong with this study into that study. And at that point, it's shared with others who have access to that centralized study repository. Collaboration tools. So like I mentioned, there's a lot of things kind of required to make these projects work that go beyond just the data itself. And so we've got a variety of things like wikis and message boards, issue trackers, just kind of a generic file drop spot. All of the things that I mentioned here are being full text search indexed. So kind of Google style searches over that. You upload a PDF and you can find participant ID in that just from the search box in the upper right. We also have what we call lists, which are basically a way to easily create a table in the database, but just as an administrator, as an end user of the system, so not having to know SQL or have direct database access. So being able to customize to configure to capture data types that we don't have maybe a full-blown module. So kind of similar to what I was talking about with that general purpose assay, where you kind of define, here are the things I need to keep track of for any given piece of data without having to go to kind of full-blown development. Security and permissions. So like I said, this is kind of a fundamental thing. It's something that we realized early on with that proteomics installation 15 years ago or so, that different people in the lab had different levels of access. You want to share certain parts, but not everything with a collaborator. Maybe it's within your organization, maybe it's external. And so we've kind of made sure that that's through and through part of LabKey, as opposed to kind of added as a shim layer on top after the fact. So we use what we call a role-based security model. So basically you're assigning users or groups to roles. So a role might be reader. Basically you can view the data, but you can't change anything. Or you might be an author. Basically you can kind of submit data and edit your own data, but you can't change stuff that other people have uploaded. You might be a full-blown editor. You might be an admin, where you can change permissions for other users, create subfolders, and generally do anything. Again, we're auditing those changes, so we can go back and say who did that, and then when you find out it was you, I'm sure I had a good reason. When was it? Kind of dig back into that history, kind of understand how you got to that current state. And then these are generally assigned on a kind of a folder by folder basis. So generally, kind of create a separate folder for each, say, major study or each data type that you're working with. This is kind of an area that we often are working with people on best practices on. How do you want to slice the data? Is the primary thing that all of this is an influenza study, or is the primary thing that we want to have all of our flow data in one spot, and all of our Luminex data in one spot? And kind of what are the different ways we want to kind of slice across those different dimensions to be able to extract it. Reporting, generally speaking, so there's kind of a bunch of different levels here. So a lot of the modules have their own set of visualizations or reports kind of predefined, where they know what the data represents and interesting ways to view that might be of common interest. We have a variety and growing list of kind of chart and other kind of configurable report types. We're not trying to, say, compete with GraphPad or have the full flexibility that you could get doing custom development in R, but kind of Excel style plots where you don't have to export or integrate with another tool. Just kind of click, click, click in the UI. Great, that's useful. Save it. Let me share that with other people who have access to the same data. And so the two examples, obviously kind of a scatter plot and line plot. I think that one happens to be a time course of data across multiple participants. Each participant there is a different series in that line plot. So kind of trying to expand the number of cases where you don't have to go to some external thing, but not trying to be exhaustive either. And then kind of going beyond that, so if you want to have more control, more customization, being able to write custom SQL queries over that underlying data, being able to plug in your own R scripts or JavaScript to produce your own visualizations and have that be interactive for the user. And then, of course, we're always understanding there's no way we're going to cover every use case. Make it easy to get the data out in either an API format or just kind of file export to be able to use and leverage elsewhere. Connecting to external systems, like I said, we're very much, how do we mix and match and complement with other systems that the groups already have or fit pretty well. So one major category is kind of database level connectivity. So running ETLs or extract transform loads. So we want to take data from some source system and pull that into LabKey database where it's more easily connected and integrated with other data. We can also just do a live view over some external data source. So if you have, okay, there's a schema in MySQL over here we've got another application that's already populating and maintaining that. We can expose that within LabKey in a secure way, and not have to make a separate copy on our side. And so, like I said, that can kind of go beyond the Postgres and SQL Server that we use as our primary data store for kind of really the main set of database platforms out there. And then we also integrate kind of specifically with individual tools. So Freezer Pro and RedCap are examples of kind of third-party systems that we have, custom modules that basically leverage those APIs in those server packages to extract data and kind of mirror that on the LabKey side. So generally, we're doing that kind of as a one-way transfer on a nightly basis or whatever the frequency of data changes is. And then a lot of times people will use LabKey as kind of a distribution point for that data. So you might not give your collaborator direct access to Freezer Pro or some other LIMS, but you can see the data on the LabKey side. So there's kind of a pretty wide list of things that we have connected with, and always kind of working with groups either to help them develop kind of a direct connection, or kind of adding that as part of our development effort. APIs, so being able to kind of enable a wide variety of development internal with LabKey, Binnell and others doing that work, or external developers kind of building on top of the data that's been imported. So we have kind of bindings for kind of some of the most popular languages out there that people are using in this space. JavaScript is probably the most, I'd say, complete of those, because that is what is driving a lot of the custom user interface in the web application itself. But R, I think, is really one of our main areas that collaborators are creating their own scripts. That can either be a script that runs external, so we have our LabKey package, which is in CRAN, that you can use to connect. You're always connecting in the context of a specific account, so security and permissions and roles associated with that account. So it's not a way to gain access to additional data. It's just a different way to leverage it than through the web interface itself. So you can either connect external, say from your desktop machine or some other RStudio install, for example, or you can also give LabKey server your R script and say, surface the results of this script, whether that's a plot, just a PNG or JPEG, a PDF, or similar, and make that available as an alternative view within LabKey. So basically, anytime you're looking at a grid of data in LabKey, so you saw some of those screenshots, you can sort, filter, etc. within the UI. You can pull in additional columns or hide columns in that grid, kind of related data. You can export that, and you get an Excel file that basically matches exactly the grid that you saw in the interface. Or there's an option to do export script, and you get a little chunk of R code that you just copy paste into your own R script, and it can connect and get a data frame that looks exactly the same. And so similar, we can also take that grid of data, hand it to an R script as its input data frame, and then show that result in the web interface. And so the end user doesn't have to know anything about R. They don't have to know it's R. They can just choose that alternative view and see those results. And so that's really enabled a lot of things for the ITN example I mentioned before, for the Hip-C immune space. And there's a number of views in the EHR here at the Primate Center to do, for example, a pedigree plot of a selected animal and its relationships within the colony. I know some people had mentioned being interested in, okay, what are some of the recent things that have happened, kind of new things. So this is not by any means an exhaustive list. You can go to see the release notes on our documentation if you want more details, and but some of the highlights. So kind of our cadence is we release every 4 months, so 3 times a year. And our numbering scheme is the kind of major version number is the year of the release, and the point after that is the index of that release in that year. We are one-based, not zero-based, which still drives some of us crazy. But we, so kind of our 3 releases this year, adding new column statistics. So we, for a long time, could click on any column in one of these grids, see what's the min, max, average, etc. So we added mean, median, or already had mean, median, quartiles, etc. So kind of quick access to some of those pretty basic stats. New capabilities within some of the built-in plot types that you can just configure point and click in the UI. Like I said, kind of a major focus in QC, so being able to do longitudinal tracking of mass spec data. In fact, we even have an auto loader so that as new QC samples get acquired, as soon as that raw file hits the disk on the instrument, there's basically a utility watching for that, running analysis, importing that, and so within 2 minutes of that acquisition being complete, you've got new data points, and doing Levi-Jennings and other kind of process control style reporting on that to say, oh, your instrument is out of control, you need to go back, and you don't want to run any subsequent actual experimental samples because your results are going to be trashed. 17.2, so our most recent release, kind of the official current version, adding more kind of permission options. So users defining views, you used to have to be basically an editor to be able to share a view with other users. So you built some custom view and you want to expose that. So we had some new use cases where people want to be able to create these views but not have permission to edit the underlying data. And so kind of breaking that out as a separate category. And so we're constantly kind of adding kind of new granularity for these different usage scenarios. Being able to, during assay import, so that general purpose assay that I mentioned, where you just kind of take a TSV and import that without the server knowing too much about it. So kind of adding more capabilities to know a little bit more about it. So, okay, this particular field is pointing at a sample ID, and so let's automatically link that, associate it with the run, wire that into kind of the hierarchy of lineage and usage, and kind of not have that be a separate manual step. Stuff that we're working on right now for our 17.3 release, so about 2 months off, kind of revamping the overall kind of frame and user experience in the UI. The header is quite different, so that's been kind of a major effort, updating to Bootstrap and some other kind of more modern JavaScript and CSS libraries, adding the ability to create line plots within the UI as opposed to just being able to do scatter plots initially. Working with a variety of different groups on kind of compliance-oriented features. So CFR Part 11 compliance, so we're adding ability to do electronic signatures. Working with a number of groups who have recent requirements to be FSMA compliant. So for those who not familiar, kind of operational, kind of best practices, system security, et cetera, audit tracking, making sure we've got everything assembled there. Just added the ability to use Markdown in wikis as kind of the formatting rule. So we could do just kind of simple text or wiki format or HTML. Now Markdown is a supported kind of markup language. And so that's about it. So kind of next steps, if you're interested, obviously I think there's kind of a variety of different levels of use of LabKey so far in this room. Some like Daniel who've been involved for years, other groups that are leveraging it already. Probably some of you, this is kind of the first time you've heard more than 3 sentences about it. So take a look at documentation. LabKey.com is more kind of the company site. LabKey.org is more kind of documentation, support, kind of usage of the software itself. Daniel's been kind enough to kind of help coordinate this session, and I'm sure would be happy to connect people on an ongoing basis here. It's open source. You download it, give it a try. We're in the next couple of months rolling out a new option to kind of on demand, just got a hosted instance because the setup, it is a kind of full-blown server installation process, database, web server, et cetera. So we've got a Windows kind of just GUI installer. People want to play with that, but trying to make it even easier for people to kind of just kick the tires. And then we're, LabKey, the company, happy to connect. We offer paid support and training and things like that. And then happy to either support development groups like yourselves directly, kind of things you're doing in-house, or have us add new features to the software itself, and then hopefully be able to share those with other users and make it on the list of features for next year. I think that takes me to the end of my kind of presentation section. I think food just showed up. Happy to take questions now, or I think kind of envisioning a little bit more kind of informal, kind of conversational approach for the second half. So hope you stick around, and thanks for coming.